So the general objective of this courses is to make you aware about the importance of safeguarding and evaluating our genetic resources. There are some historical examples that define the importance, or they were aware about the importance of such genetic resources. For the first one is Vavilov. And then for me, as agronomist, I was also under the impression of the book of Rodney Harlan, Crops and Man, and I advise everybody to read it. It is a very important book.
Mark,
yes?
Sorry to interrupt. We still see the first slide. So you're not changing the slide, at least on my side. I don't see the changes.
And like that note. Bon, I go back, I think. Je vais sur partager. Et
voilà. Il faut que vous choisissiez l'écran dans lequel vous allez partager. Donc, si vous mettez en full screen ici, là, vous n'êtes pas encore ce petit bouton ici. Si, vous êtes déjà dessus. Ah, voilà, maintenant, vous devez choisir cet écran-là. Donc, si vous cliquez sur Zoom, là, vous devez partager l'écran. Ouais, ça sera 2. Ouais. Ok, vous partagez. Voilà. Et maintenant, vous êtes capable de... Donc, nous, on voit ça, nous.
Ah, c'était plus simple. Okay. Do you see the slides moving?
Perfect now.
Okay.
Yeah, screen.
So thank you, Baptiste. So I advise really everybody, scientists, young or older scientists, to have the opportunity to read this book of Harlan, who give us the basis of the importance of plant genetic resources. Then, in the framework of the international treaty and FAO, there are different plan of action for each country to define priorities and strategies for collecting, safeguarding, and utilizing genetic resources. So, why collecting and why safeguarding genetic resources? Because the adaptive capacity of life depends really on biological diversity. And also our agriculture depends on a reserve of diversity. And also our human being is depending on biological diversity. Therefore, plant genetic resources are really a reservoir of diversity and is a guarantee, give better guarantee to have capacity of adaptation of our human being. Also, not only genetic resources, but also ecosystems diversity is important, where we can find interspecific diversity or into the species diversity, but also cultural heritage, what was used, what are the basic recipes for using specific plants. All those aspects are very important for food security and for the future. So what is genetic resources? We could say that it is material, plant, animals, or other organisms which could be have a value as a resource for the present or for the future generation of people. That's also something quite interesting to come back to the roots, that if mankind are on the earth since 2 million of years, 99% of that time, the mankind was hunter and gatherer based society. So it was not agricultural society. So it is only the last 10,000 years that agriculture started. And if we consider that 80 billion men and women have lived on Earth since the appearance of the human race, 90% have been hunter-gatherers. So our agriculture is very a young way to life on Earth. So therefore, we have to remember that our capacity of adaptation is nowadays based on agriculture, but it was not always the case. Since the time, there is a large erosion of diversity. There is phenotypic diversity on all plants that you can imagine. There is a difference between phenotype and genotype. The phenotype is the expression of the genotype, but under the environment pressure. So it is the combination between genotype and environment. But the genetic diversity is the heritable variability of plants or other organisms, and is also the base selection for crop improvement, and it is really important for plant breeding. So when you start from primitive forms of wild species or landrace of traditional varieties, you have generally a large diversity of genes and capacity of adaptation, and or more you are developing modern varieties, or less you have the diversity. So there is a erosion of diversity. So from here it is to illustrate these things, from White selection, you reduce the genes and the alleles present in the crop. And here, if you see diversity of genetic resources, and cultivars have a much narrower genetic basis. As you was mentioned in the film, more than 95% of the orchard meadows of our country have disappeared from the time to time. So you see here the erosion of such cultivation systems. If we have to compare, so this old way to grow fruits, it was agroforestry. It was always association between trees, animals, grass production. There were a larger diversity of cultivars, low inputs, high biodiversity, rich agroecosystems, but lower food production. In contradiction with this system, you have the modern apple or pear production with high densities, dwarfing rootstocks. It is quasi monoculture and very low diversity. What can we get from these old cultivars? Very often local adaptability, local and diversity uses of such fruits, rusticity, generally pest and disease tolerance, phenotypic diversity, and so on. And when you go in the shop, you see a very small diversity. So nowadays, 5 commercial cultivars represent more than 50% of the production. And if we see inside the crop of apple, the genetic diversity is very low also. If you see, Golden Delicious has been very intensively used as parents in many crosses, but also Cox. And then you got Elstar. From Elstar, you got a lot of cultivars. But if you see the genetic diversity, it is very narrow. Continuing as an example, in Belgium, it seems to be the first biggest producer of pear, and 91% of the pear production of Belgium is based on one cultivar. So it's really a monoculture. And if we see the dependence of apple production to fungicides and pesticides, you see that here, apple represents 40 times praise, potatoes 17, and if you see mice, only 2. So you see that apple is very dependent of pesticides. Therefore, in our research program, we are aiming to enlarging the diversity, to look for low input farming systems with looking for higher resistance or tolerance of the plants to pest and disease, but also having better efficiency to nitrogen, to have plants who have a better flexibility and to be better adapted to climatic change. But we want also to improve quality and to offer healthy products and also develop more participative approaches. If we can compare, it's not perhaps the good example to use race cars, but formerly the breeders were looking race cars, and nowadays we are looking more robust vehicle to be more adapted to climate change. So if we come back to the genetic resources, we can see that genetic resources cover a large part of diversity from crop-wide relatives, ecotype, crop land traces, modern cultivars, obsolete cultivars, breeders' lines. That's mostly focused for food and agriculture. The right part is mostly used for non-food utilization. So we also consider for fruit cultivars that there are two different families. The one we could say old amateur bred cultivars, so before commercial breeding, and land traces or land or cultivars coming from the countryside. So if we can see how to define what are amateur bred cultivars, the place, the names, and the breeders or the funders are published. We know it. The names is defined with high lexical style and very often connected to the breeders itself or to his family or to a historical person or even. So only or with the quality of the fruits. Only when you see the name of a cultivar, you can have a signal, an indication to know if this cultivar is from amateur bred, from historical amateur bred origin, or eventually from more like a land trace. Then for such type of cultivars, we can find in the literature the descriptions, synonyms, historical description, and so on. And we have so many information from the literature. Very typical also for such cultivars, they were cultivated on dwarfing rootstocks and very often as espalier. And they were mainly also often collected in international collection. So mostly used for luxury fruits. And here you see a very nice description of such cultivars, old pear cultivars. Then we could also make a proposal to make a classification of old, but because what is old? Everything is relative. So what is nowadays cultivated in 1 century will be defined as old. So we tried to make 5 categories. Ancient means cultivars mentioned in the literature before 1760. Very old, between 1761 to 1850. Old, between 1850 to just before the First World War. Pre-modern, it is just after, so between the two wars and up to the Second World War. And what we could say modern is cultivars raised after the Second World War. So the local landrace, so from the peasantry, there we don't know the breeder, we don't know the place, we don't know the dates. The names are really very often in simple language or even in the dialectal language. Coistres, Poire de Fer, that's all French name, but I'm sure in your language you have such simple name. Those cultivars are not recorded, or very often not recorded in published work, so there are no description of reference. They are not found in commercial document, catalogs from nurseries or so on. They were not grafted on dwarfing roots, but on seedlings, and they were used for orchard meadows as standard trees. And for pear, specifically, they were grafted on the dwarfing rootstock quince. And very often they were selected for robustness traits. Sometimes you can get cultivars and it seems to be a landrace, but with the time you can discover that eventually it is a bred cultivars, but a change of name or the name was different. So there are many local landraces that are only old, less known classical cultivars. So it's more subsistence cultivars. It was more for food, not for looks. So landrace have a local name. There is a lack of formal crop improvement. They are commonly characterized as with specific adaptation to the place where they were born. They offer often tolerance to biotic and abiotic stresses, and they were associated with specific uses. So that's an example. I go back. So that's the first pomology which were dated in the Netherlands in 1758, where you could say already description of cultivars, quite nice described. And also Cocorico, Belgium is regarded as the first pear breeding place. Nicolas Ardampont seems to be the first pear breeder in history. And also we developed in 1 century more than 1,000 pears, amateur bred pears, and we were focused on soft-fleshed melting pears, so beurre types. So really dessert fruits were much improved in Belgium than in any other countries during 1 century. So we started here in Belgium early 1975 to collect material, and we were in much contact with the public. It's thanks to the public and the medias that we could have the opportunity to collect this material. It is only because the people offered us. You have seen all those letters that we received. It was the good old time when we could receive letters and not emails. And then we contacted such people. We went to their garden or to their orchard and collecting material. So nowadays, currently, we are still receiving several hundreds of samples of fruits each year from people sending us or coming to us, bringing cultivars, asking us which is the name. And in this diversity, perhaps 1 to 5 cultivars will continuously will be collected each year. But so we are collecting less cultivars now than in former time. Some example of diversity of apple, of pears. The pears have larger diversity of use and of forms and on taste than for apple. Here's a nice diversity of pears. So the global importance of genetic resources. It is important to come back to our roots and to look for higher robustness, higher tolerance to stresses, better adaptation to pest and disease, because they were released before plant protection products were used. So the first plant protection product was copper, and it was used as the Bordeaux mixture at the late of the 1870s. But also they selected long keeping ability without cold systems, and for sure, for us, the most important, they are expressing a large diversity of traits. Then each of us, if we are starting such enterprise, the first step is collecting. So collecting, but collecting what? I think that each of us have to define our priorities case by case, and taking into account our own objectives, the budgets that we could get on long-term budget security, and also the facilities that we have. Do we have land sufficient or not? It will define which type of material that we have the possibility to collect. We defined our priority as collecting material from Belgian origin, grown formerly in Belgium, or very old cultivars, so before the fungicide use, or local and regional land traces. And then we are collecting oral tradition and uses. And also the objective is to have the largest diversity. So we are not a work collection. So a breeder will collect material, but only for its own objectives, and with a short-term objective. We have a long-term objective. So therefore, we want to keep a larger diversity than in work collection. Then we are looking for cultivars that are expressing robustness, tolerance to biotic and abiotic stresses, and also some outstanding traits, aroma, taste, and so on and so on. So here, the key element is the people who knows the origin, the use, and so on. So we need to collect the ethnobotanical information, dialectic names, the uses, and so on. Then on place, we are taking as much as information as we can. So making pictures, collecting budroots, and also filling passport data. Here it is an example where we give where we collected, what are the specificities of the fruit, what was the use of the fruit. All those information will be used for introducing in the database as passport data. At the FAO level, there are several templates where we need to fill in all those passport data in an harmonized way. So you see here accession name and pomologic name, consensus name, and so on. There are a lot of traits that we need to collect as passport data, also plant use, tree form, plant use, etc. So here we are. The first step was collecting. The next step will be to propagate. So you collected budroots, but then you need to propagate the material. And for fruit trees, the only way to propagate material is the vegetative propagation methods. Here you see to graft the summer grafting. So you have to take off one bud from your bud wood, and then you insert your bud between the bark of your rootstock. You ting it, everything. It stays the whole summer as a dormant bud, and in spring you are cutting just above the bud your rootstock, and your tree will raise from one single bud. Then importance is the traceability. So when you are collecting material, you give to this accession a unique identifiant. So here it is 1202. That's the unique identifiant which will follow during the whole process this material. Here we are developing a specific label. It is a metal label that you put it in spiral around your rootstock. And here we defined which rootstock we are using. Mostly we are using M9 because we spare time and space by using dwarfing rootstock. We have our own experimental nursery where we are propagating all our material. We need sometimes so to take into account some incompatibility with some rootstocks, specifically for pears. Many old land traces are completely incompatible with quince rootstocks. So that's constraints with pear. Then you raised your trees. Normally we are doing 5 trees per accession. 2 trees will go. Sorry, I've no. Okay, 2 trees will go to such evaluation orchards. 1 tree in an evaluation orchard, and 2 trees in repository orchards. Where is my thing? Sorry. Here. So you have then 2 types of conservation. You have an ex situ repository orchards. That's here at our institute. There we have 1 tree. We have 2 trees for each accession in the repository orchard. So 2 trees there, and 1 tree planted in the evaluation orchard in non-sprayed evaluation orchards where we will develop first and second screening. And then afterwards, but that will be at the end of my presentation, we are developing a network of repository orchard, but on-farm for on-farm conservation. I come back to the propagation. We have the trees, we have planted the trees in the evaluation orchards. Here you see how it was when it was young. That is how it is today, because I took this picture today, so the trees are quite bigger and you don't see anything anymore. And here I come back to the process of evaluation and also the importance to work together at the European level under the umbrella of the European Cooperative Programme for Plant Genetic Resources, where we developed a common project for standardizing the protocols and tools for characterization and evaluation of our fruit genetic resources. So ECPGR is a collaborative program among most European countries, so it's larger than EU. And the aim is to ensure long-term conservation and facilitating the utilization of plant genetic resources in Europe. So there are 38 countries. There are different working groups for each crops. And here our specific objectives of our work was to edit ECPGR methods and description list for the characterization of the evaluation of apple and pear genetic resources, because there were many different descriptors for different users and goals, but we need common protocols and methods when we want to compare and to share our results. And therefore, also we need specific evaluation descriptors. So we published one now for 2 years ago. Those descriptors are already available for everybody, one for apple and one for pear. The first important point was to define common reference cultivars. Reference cultivars are very useful for having comparative results and to compare our material, unknown material, with well-known material. And the idea is also to have in each site, at least not perhaps all the list, but some of them need to be present in any place in order to be comparable with the different places and the different institutes who will develop assessment work with genetic resources. So that's the list. That's an example. The list of that we defined for apple, looking for apple that could be grown in the north of Europe, but also some cultivars that could be grown in the south of Europe. So we need a larger scope of cultivars. Very important was also to harmonize the phenology stages. There are different ways to follow the evolution of phenology stage. From Fréquingé, it's a French way. CRA, we developed our own way, but the most important way to follow the phenology stage for each crop, in fact, is the BBCH codes, and it is an international code, and we are following formally currently this way to characterize the evolution of the phenology stage. So very important is to know the flowering phenology and to classify our cultivars in different flowering seasons. This way of classifying cultivars can only be done after several years of observation. So you need to follow your cultivars at least during 6-7 years before you can classify them in the 9 places of flowering season. And also we need to follow the flowering intensity of the trees, because when trees have very low intensity of flowering, it has nonsense to record the flowering phenology. Then very important also, I take some example of evaluation traits. I will not go for each of the traits, but I take only some example. Here it is the same principle to classify the fruit time ripening of your cultivars with the importance, and here I come back always this importance of the reference cultivars for the phenology of flowering and for the phenology of ripening, because then you can compare your cultivars with reference cultivars, and in fact all our data will be data that are relatives, because it is all time adapted with the environment where you are. Here you have more characterization data. So the fruit shape, that's a simple way to give fruit shape. There are more sophisticated system to classify the fruit shape, but mostly those 5 types are sufficient. Then fruit size, it seems to be important for many people to have the good to classify. Also, you need to do your assessment during several years, and then to have an average. Then you can work on weight of your fruit or on diameter of your fruit, and then you can classify your cultivars by fruit size. That's also a characterization trait. It is the crowning, how the fruits are crowned at the apex is very important trait for the cultivar names. It helps us for classifying the cultivars and looking for the names of the cultivar. It's not very useful for the utilization, but it is more for the characterization of the cultivars. For me, important is also the fruit bearing habit. So the tree architecture is a very important trait for the utilization of the cultivars, but also for breeding. Knowing the fruit bearing habit is very important because it can reduce the fruit biennial habit, so production 1 year on 2. Then I come back to the last description of evaluation protocols and methods, and it is really the key work that we developed here in CRAW is developing way of assessment for disease and pest susceptibility. So we are more focused on disease than on pest, but pest is becoming more important. So we will also develop pest assessment scale. So here we need really to have a long period of evaluation, because the 1 year you have good conditions for the development of one disease, the another year for another. And so you need a diversity of climatic conditions to be able to evaluate properly your material. Ideally 5 years, but more is better. And if you want to make a proper disease evaluation or pest evaluation assessment scale or assessment level, you need to don't spray your orchards. So we are doing evaluation since more than 40 years, and we never sprayed against pest or disease. So that's for us a key element for having a way for good evaluation. Then we need also good reference cultivar. For example, if you have high susceptible cultivars, Gala, Golden, Delicious, Crisp, Pink, so on, you will see the pressure of the disease of the year. If you have no disease of Golden 1 year, it means that this year there was very, very low scab pressure. So it means that your information will be of poor quality. Then an important point is to check if your pressure is homogeneously distributed inside your evaluation orchard. Otherwise, if you have cluster of pressure of infection, all your material is not evaluated on the equally conditions. So that's also important to see and to check how are distributed your disease or your pest in your evaluation orchards. Then an important point for the disease assessment is the fact that in phytopathology, the intensity of the disease is the sum of two components, is the sum of the incidence and the severity. Incidence is a qualitative presence or absence of symptoms. So it is generally defined as a proportion of organs affected by at least one symptom. So it's really a qualitative evaluation. It is yes or no. Then if you say yes, then you can evaluate what is the severity. Then it is a quantitative proportion of surface, length, or volume of an organ which are affected by the disease. And sometimes you can separate incidence and severity, but normally we are doing that on a global way. And I will go back to the incidence scale. Here, based on empirical facts, an evaluator has only the capacity to discriminate exponential differences of symptoms. Therefore, we developed an assessment scale which have an exponential proportion of infected organs. Here you can see an example of incidence for apple's cap with the proportion of infected organs with at least one symptoms. So one, no symptoms, you have to look, you never seen ever one symptoms. Two, you have to look after very carefully, and then you see, oh yes, very few symptoms, but very few percentage. Three, you see directly symptoms, but also in a limited number of organs. Then you cannot, for us, we cannot make the difference between up to 5% and more or less 25%. Therefore, we developed an intermediate rating state between 3 and 5. Then you can also make quite easily an assessment of 50% of infected organs and more than 90% of infected organs. And in between, when you are doubtful, you put intermediate rating. That's the principle of incidence. Then for each disease, there are some prerequisites. The first is you need to know the biological cycle of the disease, how the disease is expressing and how which are the cycles of the disease with the season. It helps you to define what is the best period for assessing the symptoms, so it means which is the best period for having the best way to could assess the expression of the symptoms. And also it is not really well defined in phytopathological books. Mostly they are presenting pictures of characteristics symptoms, but on heavily susceptible cultivars. And here, with a large diversity of cultivars, each cultivar will express differently the symptoms of disease. So you will be more careful to detect what are the symptoms, because it could be different cultivar by cultivar. Here you can see an example of severity, so the importance. So each of these leaves have at least one symptoms, but this leaf is much more infected than the other. So you see that it is a quantitative importance of the disease that is expressed by meaning the severity of the disease. And very often, sorry, I go here. Very often we are combining incidence and severity in one scale, because we have not so much time when you have to assess several hundreds of cultivars. Having a global assessment scale is often sufficient for having a good evaluation. And here we defined some words for having the capacity to make the difference between the different classes. But you see always intermediate rating classes where you have some doubt, and we start to evaluate the severity when you go up to the scale, to threshold of more or less 50% of the organs are infected by at least one symptoms. Then you go to the severity scale that helps to define where you are with your assessment scale. Another point also that I forgot to express when I come back here is that because we are looking for cultivars with a high level of tolerance to diseases, we are much more precise in this state of the importance of the disease than here. If you go if you are in front of a cultivar with more than 25% of the organs infected, it is already too infected for be grown without any fungicides. So it is less important to know if it is a 6, a 7, a 8 of a night than to be sure that your cultivars is classified in a 2, a 3, or a 4. There, it is very important to know where is situated your cultivar. So therefore, those scale with an exponential way of expression of symptoms is very focused on the objective to select more tolerant disease cultivars. So I come back to then here, just to express some example of specific symptoms. Not always you can find such symptoms in literature. It is Venturia pyrrhina on pear, and it is specific for pear. There are symptoms on twigs. In England, in the past, there are also description of symptoms like just like that on apple. I've seen in the literature that in high pressure of scab, you could also got some twigs scab symptoms on apple, but normally not. But on pears, yes. But then you need, for sure, having long-term non-sprayed evaluation orchards to let the disease to be spread in the orchards. Here you can see also specific symptoms for scab. No, it is on that's a problem. Sorry, here it's not on fruit twigs. It is on leaves, and specifically for pear, the symptoms are seen under and on the underside of the leaves. Very often only on the underside in the early stage, because at the autumn very often you don't see them anymore. So the importance for pear is to do your evaluation not too late in the season. I would suggest by the end of mid-June and June. It depends on the season, but not later. Otherwise, you will not see any symptoms anymore. So just from an example. Then we tried also to look for assessment scale of robustness trait. What is robustness? We will have probably also in the next future a lecture on what could we call robustness. We made also a survey about who is or how we can define robustness, because it is a large terminology. It is a capacity of adaptation to biotic or abiotic stresses. And here, one of the way that we developed was the assessment scale for global tree foliage health. Here it is also a more exponential scale. And here we have an example of how we could define the quality foliage of different cultivars. And you can see that there are really cultivars with a better foliage quality than others. And it means that those cultivars have either a better adaptation capacity or either a better nitrogen efficiency. It's a complex trait, but it is important for us for defining where our cultivars are in this scale. We can also develop other description, like here, sugar content, C vitamin contents, and also that's more for the characterization. We developed a microsatellite analysis of our collection. We were surprised to see that many of our collection are triploids. I think more or less 30% of our cultivars are triploids. And also we see what are unique, what are duplicated, and also compare in comparison with different other countries. Then we need to mention that the molecular markers will not do any difference between the mutants. For example, Redhead du Canada, you have the gray form, so the russet form, and the non-russet form. The molecular markers will not make any differences between the two. So you need to collect phenotypic data to separate the differences between those two cultivars. The same for Cours Pendu. You have a gray one, and you have a pink one. So if you have only molecular markers, you will not make the difference between the two. It's the same with Eijdener Klumke. It's a Netherlands cultivar. You have a red one, but you have a striped one. They are different. So mutants are very important for fruit cultivars. And also there are population subtypes. Here you see the diversity of expression of the phenotype of a very old landrace of our country, large mouche, which has several synonyms. The molecular markers will also never see the difference between all two, between a much striped or a much less striped, or they are also more early and more late. So all those traits need to be phenotypically evaluated. And then my last 2 slides, we were aware that we collected from the countryside all those cultivars. We collected them, we propagated them, we put them in the ex situ repository orchards, we put them in evaluation orchards. But the fact is we concentrated everything in one place, and that's really dangerous. Therefore, we developed a strategy for long-term conservation where we decided to repropagate material and to plant back where the cultivars were originally raised, and to develop a network of repository orchards in different parts of our region. And in different geographical regions, all regions need to have also their own repository orchards. And for us, it is a way for having a long-term safe conservation of the diversity, and also to create a more dynamic evolution of the diversity, not only keeping the material ex situ, but to come back to a form of on-farm conservation and multi-local conservation. And all those repository orchards have been grafted on seedlings. So it means there are orchard meadows with animals and fruit trees together. And we already could propagate 1,200 cultivars of all species, apple, pear, plum, cherry, on those repository network of orchards. Here you can see an example of such repository orchards. And in conclusion, we need to identify and to conserve the large diversity of phenotypic, but also of genetic diversity. It is a cultural heritage. It is also a traditional knowledge. And also we need to develop safe conservation strategies for our genetic resources. For us, the only way to have a good way to use the material is to develop long-term evaluation activities in non-sprayed orchards. And we are looking in these conditions. It is the only way to have a look in these conditions for tolerance, resistance to pest and disease, robustness, biotic and abiotic stresses, but also healthy and quality traits, and so on. And that was my last slide. So I really thank you for your attention, and we are open for any questions, but I think I was perhaps too long in my presentation. So thank you.
Are there any questions? Anybody?
I see Philip and Inde.
Please, Philip.
Good afternoon, Inde.
Yes, thank you for your presentation, Mr. Lateur. I really enjoyed it. I just want to throw in some thoughts. You were talking about local adaptability, but when we talk about local adaptability, it always refers to past conditions.
Yes.
When we think about local adaptability in the face of climate change, it's when we have a region with a local adapted cultivar, it's maybe not so interesting for the future. So would it also make sense to look in other regions for local adapted cultivars with another climate?
It's a good question. It's a very good question, because it is often a question that I received. In fact, when we say local adaptability, there is a diversity of conditions where the plants were grown in the past, and therefore there is a diversity of place and of soil and climatic condition. Even in a small surface as Belgium, you could not imagine the diversity of pedoclimatic conditions could be different between the north of Belgium, where it is a flat, and the more higher, not for Switzerland, because the highest point of Belgium is 600 meters. It's not like France anyway, but there is quite a large diversity of conditions where the cultivars were adapted. And the second point is we need to look the largest diversity of traits. And even in a small country as Belgium it is, we have found a large diversity. Then do we need larger diversity than that what we found from the past? We don't know. We really don't know. But perhaps that we need some assisted migration from surroundings countries, not too far, I think, because otherwise we have too many conditions that are different. But we probably will have to adapt us if needed. But thank you for your question. It is an important question.
Thank you.
Inde?
I didn't have a question. It was clapping my hands.
Thank you.
But you have there were one thing, but my English is not so good. It's an adaptation to drought.
Yes, that's
it's a question of
that's also a good
yes.
question. The biggest advantage of ex situ collection and ex situ evaluation is that you are evaluating all your material in the same pedoclimatic conditions. So you bring together everything in one place, and you can compare everything in the same conditions. Otherwise, you are not able to compare one cultivar in one site and the other cultivar in another site. The environment is so different that you cannot compare the cultivars. So therefore, having planted all the cultivars in the same place, in the same evaluation orchards, when there are drought conditions, so 2 years ago it was very drought, or 3 years ago, I don't remember, then in the same conditions we can compare the capacity of adaptation of the cultivars for either drought condition or wet conditions or heavy infection of scab or canker, I don't know. But we cannot do better than that. The next step is for reducing number. Yes, also an important point is when we are doing evaluation with a large diversity of cultivars, so we have probably 500 or 600 accessions in the evaluation orchards, we are doing a first screening. We are not doing a proper evaluation because we have no sufficient replicates, it is not randomized and so on. But we are doing a first screening, and the best that we could evaluate or point out those cultivars, then we can make a new evaluation with more replicates, but with a reduced number of cultivars. We can make a better evaluation orchards with replicates and randomization block with 20 cultivars. That's feasible, not with 500 cultivars. And then we can also play with the rootstock to compare. For me, the better adaptation of our fruit trees will be to play with the rootstock capacity of adaptation with the drought conditions, because rootstocks, we know they have the different rootstocks have different capacity of adaptation with their roots. Some are adapted for sandy ground, other for more heavy grounds. The surface of exploration of the roots is different from one rootstock to another. I think the future, the choice of rootstock will be more and more important for organic fruit tree production. But that's my opinion. Thank you, Inde.